Hi everyone, welcome to Reason with Science. I'm your host Jitendra. This episode is with Nick Lane. He is a professor of Evolutionary Biochemistry at University College London. He is an author of several books like Power, Sex, Suicide, Life Ascending, and The Vital Question. His latest book is Transformer, The Deep Chemistry of Life and Death. Here we talk about what is life, life as an information, importance of Krebs cycle, how did the life start, consciousness, chronic diseases like cancer, and process of aging. Enjoy the conversation, share and subscribe to support the podcast. Thank you for listening. Hi, Nick. Welcome to the podcast.
Thank you. Pleasure to be here.
Yeah. So once we think of, having a conversation about origin of life, first question that always comes, what is life? Right. Thinking about this question, and of course, there is a bit of history about it. There are 4 books written so far on the question.
Entitled "What is Life?"
Exactly.
You've been counting, have you? Right.
Exactly,
I know probably 3 of them. I'm not sure about the 4th.
There is
yes,
one from chemist Adi Pross.
Yes, I'm familiar with that one, and Schrodinger's famous book, and there was one by
Exactly.
Haldane as well.
Yes,
which turned out to be a collection of essays, which was slightly disappointing.
Exactly, and the recent book by Paul Nurse, What is Life?
Oh, of course, yes, yeah. Right. Oh, very good.
Yeah.
I still think it's the wrong question.
That's what my question was. What do you think? It's a good question or not?
No, I think it's an unanswerable question. I think it's really the wrong question, because what it makes you think about, as soon as you say what is life, we tend to think of an object, of a thing, of a person, of a living object, you might say, rather than as a process over time. And we are living our lives and we're breathing continuously, and we'll be dead if we put a plastic bag on our head and so on. But if you're thinking about the origin of life, then we need to strip away all this complexity. And all the cardiovascular system and respiratory system that's supplying oxygen right to our cells, and realize what that is a continuous chemical reaction between effectively the hydrogen stripped out of food and the oxygen that we're breathing in, and it's powering everything that we do all the time. And so this is the process of living over time. And if we're thinking about the origin of life, then we need something comparable to that, but stripping away all the complexity of the cardiovascular system and everything else. But we still need a continuous chemical reaction. And so we're looking, I would look for places like hydrothermal vents, where you've got a continuous flow, a continuous reactivity, a continuous bringing together of effectively reactive fluids, which is in some way analogous to breathing. So the idea then, we've stripped away pretty much all the thing that you would call life, which is to say the organism, and we're left with a reaction, a continuous chemical reaction. And that, from the origin of life, starts with something very, very simple. We're starting with prebiotic chemistry of the simplest kind, of hydrogen reacting with carbon dioxide to make small organic molecules, Krebs cycle intermediates, and this kind of thing. And we have to go from there all the way up to genes and coding for molecular machines, and a tremendous amount of complexity. And that's a long, if you like, evolutionary distance. It's a long distance from the simplicity of prebiotic chemistry all the way through to the complexity of a living cell that we would recognize. And it's almost impossible to draw a line across that at any one point and say, now it's alive. Without all the preceding stuff, it would never have come to being alive. And without, I would say, the faster we can get to information, if you like, the better. And if you wanted to draw a line across, it would be the introduction of information into biology. But it really matters into what context do you introduce information, because we start out with no information and we need to end up with information that has meaning. And so it's an arbitrary place to draw the line there as well. So any question, what is life, tends to implicitly suggest that there's a moment, that there's a kind of a phase transition, that we go from a non-living thing to a living thing. And that in some way it's an object rather than a process. So I think the question it's a really human question, and we all want to ask it, and it's a great title for a book. But I think in the end, it's just fundamentally misleading about what we're trying to answer.
Yeah, I mean, of course, once we think of this question, the information part, it generally comes in. Uh, I think it was already introduced at the time in the last century itself, especially with the code of life, with the structure of DNA, etc.
Well, the famous book by Schrödinger, What is Life, was really, this was written at a time, in fact, when we did know from Avery's work, this was in 1944, and Avery had just published that the hereditary material is in fact DNA, but not very many people believed it. And Haldane, who also wrote a book called What is Life a few years after that, had dismissed DNA as being relevant to hereditary material on the grounds that there wasn't enough variation in its chemistry, and he pointed to histone proteins instead, as proteins generally as having much more variation in their structure and therefore much more likely to be a code. And he kind of missed the point. Schrödinger followed Haldane's view and was also thinking about proteins rather than DNA, but he talked in much more abstract terms about an aperiodic crystal, which is to say some kind of crystalline structure that doesn't have a completely repeating structure to it, where the intimate structure keeps changing. And actually, he was really describing DNA and the letters along DNA without knowing that was what he was describing. This is one of the wonderful things about that book, is he has an abstract view of what's required, and actually now you read it now, you realize he's describing DNA, but he didn't know at the time what he was describing. He was simply saying, this is what it must be, something like this. So that's why it's still worth reading this book. But we've had an obsession, I would say probably a healthy obsession, with DNA. It's obviously incredibly important, and it's taught us a tremendous amount about the way that life works, but it's tended to marginalize other important aspects of how life works, including, from my own point of view, energy flow and metabolism. And we tend to just assume, without really proof, that information, that genes invent metabolism and invent the way that cells operate with their energy systems and so on. And there's a lot of serious problems with that view. And if you think about it the other way around, that energy flow and metabolism came first and genes arose in that environment, that has its own problems as a view, but it also solves a lot of problems. And from my own perspective, it's a really productive way of asking these questions.
Yeah, so I mean, of course, these different schools which are there in origin of life research, what you mentioned, that metabolism first, or replication first, information first, etc., or they can be also membrane first, this kind of notions. But I mean, of course, we can go beyond that, and more research papers are coming on this perspective that it was not like only either RNA world or something, that it was all soup cocktail of different chemicals. And so out of that, there has to be something simple but meaningful which has to come out, which will give a kind of this precursor for the origin of life. And in your view, it's metabolism, right?
Yes, I would say membranes and energy are equally important. And this is how I came to the field of the origin of life, because my own background is in bioenergetics and how mitochondria work, and mitochondria are the power packs in our own cells, and it seems like a million miles away from the origin of life. But mitochondria were free-living bacteria once, and they're powered effectively by electrical charges on the membrane. And it's a very, very sophisticated system with lots of molecular machines and a membrane which is practically impermeable to protons, and protons are tiny things, and it's difficult to have a membrane that's impermeable to protons. And yet this system is universally conserved across life. Pretty much all cells work this way. And so you're left with this strange question that, well, it seems to be really important, really fundamental. It's a strange thing that all cells have got an electrical charge on the membrane surrounding them. And yet the kind of the systems that are using it are incredibly sophisticated and plainly the product of genes and natural selection and nothing to do with prebiotic chemistry. So there's to my mind, a paradox there about, well, how could such a system have got going and become universally conserved across all of life so early on? And so you start to think about, well, what kind of things is it powering today, and how does it do it? And many people would be familiar with the ATP synthase, which is a beguilingly marvelous machine. It's a rotating motor that sits in the membrane, and there's a wheel, if you like, in the membrane, which is turned by the influx of protons across the membrane. And that's making mechanical changes in the head group, which is making the universal energy currency of life, ATP. And it's a gobsmackingly impressive machine. It's iconic, really. And it's also, I think it's easy to be taken in by it. I spent a lot of time trying to think, well, what could be a prebiotic equivalent to an ATP synthase, something that doesn't need genes to, and I could never really think of anything. And then I realized it's missing the point. It is a product of genes. It came in the era, it arose in the era of genes and selection and so on. So what could natural geochemical proton gradients drive before that, and something equally important to life, which is CO2 fixation. And a lot of bacteria operate that way. They're using the inflow of protons into cells and the electrical charges on the membranes, not to just drive the energy currency of life, but to drive growth, in effect, to turn hydrogen and CO2 into the organic matter, into more of themselves. And that, unlike the ATP synthase, this exquisite molecular motor, this is, I won't say it's simple, but you can conceptually imagine what prebiotic versions would be. We're dealing with little clusters of metal ions like iron itself and sulfur and nickel and so on, and they form little mineral-like structures inside proteins today, and these same minerals will form in hydrothermal vents on a larger scale. And you think, okay, so we have an inorganic barrier with these minerals in, and we have different fluids which are mixing, and possibly the electrons or protons can cross these barriers. Can it drive work? Can it drive the reaction between CO2 and hydrogen? Can it turn gases into living molecules? Can it drive growth? Can it structure how all of these things happen? So really what I'm saying, and over the last 5 or 6, 7 years, it's gone from more or less hand waving that it would be wonderful if it did, and theoretically it should, and to it really does happen. It's being done in the lab. It's still a question mark. Can it happen on the right scale? Can it really drive everything we want it to drive? But the basic first steps, yes, it does work. And so then what you're left with is this feeling that, okay, so we've got a structured inorganic system which is capable of marshalling energy flow to drive the origins of metabolism, to drive the reactions between hydrogen and carbon dioxide to make the organic molecules that make up life. And one of those organic molecules, in the end, are the building blocks of DNA and RNA and so on. So what we effectively have is a system which is capable of growing, of converting a disequilibrium in the environment into a growing organic system in which genes and information emerge, and then they modify how it grows and so on. So there's a lot in that which is hypothesis. Let's say hypothesis, because you could say speculation, but let's call it a hypothesis because it's testable. It's not necessarily easy to test, but it makes specific predictions that are testable, and quite a few of them are beginning to look as if they may be true.
Yeah, but this is it's so interesting, since what we are discussing here is the literally gases coming together and forming life material that we even use like in modern days. But there is a lot of base to cover there.
It sounds crazy, doesn't it?
So starting with the information. So once you mentioned about ATP synthase, we can think of it as these molecular machines, again, coming from DNA. So DNA can.
So I think I would place that later as a product of genes and information.
Yeah. So the point being that I mean, of course, that so these ATP synthase and stuff that that you just described, they come from DNA. I mean, it's not about the information at the origin of life scale, but this is what these are the molecular machines that we use nowadays. So you are
Yes.
looking something equivalent of that kind of system in the origin of life scale, which would be completely inorganic. And it can build those kind of how to say energy storage molecules, because
Well,
these
it's
are
not so. I
literally.
mean, it's not so much energy storage. I mean, there is some energy storage involved, but what we're dealing with is two gases, hydrogen and carbon dioxide, and I find this strange, but under the kind of conditions that I'm talking about, that's to say 50 degrees to 70 degrees centigrade, alkaline conditions, a relatively high partial pressure of hydrogen, which is there's fluids bubbling with hydrogen gas, and an alkaline pH. So under this set of conditions, which correspond pretty much to an alkaline hydrothermal vent, it's a type of hydrothermal vent that we find on the bottom of the sea. Under those conditions, thermodynamics says, and this is a scary word for a lot of people, but basically just think of water flowing downhill. Water doesn't want to flow uphill, it wants to flow downhill. These two gases will release energy if they react together to produce methane and water, for example. But they don't react together very easily or very quickly. It's quite a difficult thing to do, and chemists have been trying to make this reaction work for a while. And it's a really important reaction because we can already split water, get the hydrogen out of water, but we can't economically make it react with CO2. If we could, then we can effectively strip CO2 out of the atmosphere and turn it into something useful, organic molecules. Let's call them hydrocarbons. Let's call it, it's the basis for all of humanity now, and it would be basically carbon neutral. But this is what life is doing all the time, and it doesn't. Overall, it doesn't cost any energy. The problem is to get them to react in the first place. There's a kinetic barrier, which is to say they're quite stable molecules and they don't really want to react. When you get them to react, then they'll release energy, but getting them to react is the hard bit. And it's the electrical charges on the membranes that are doing that. And these are provided free of charge in hydrothermal systems, in certain hydrothermal systems, and are capable of effectively facilitating the reaction between these two gases to make organic molecules. So thermodynamically, having hydrogen and CO2 in the strict absence of oxygen, this won't work if there's any oxygen around, at 50 degrees centigrade, and you shake them up, what you should get is cells coming out. Thermodynamics says cells are more stable than a mixture of two gases, hydrogen and CO2. The reality is you can shake it up for 500,000 years and nothing much is going to happen. You've got to get them to take those first steps to react together, and that's what the electrical charges on the cell membranes are doing, or on inorganic barriers in hydrothermal vents.
Yeah, but why hydrogen and CO2?
Well, that's an interesting question, because that is how life works on Earth. So then there's an interesting question. Well, is this just would life work that way somewhere else? Is there any reason to think, or could life be completely and utterly different? And my own feeling is most life that we find would not be so dissimilar to life on Earth. And that might seem like a really limited imagination, but it's based on carbon chemistry, really. Carbon is really good at the kind of chemistry it does, much better, say, than silicon at the kind of chemistry that it does. It's very good at making large, complex molecules. And they're strong, they're robust, they don't fall to pieces very easily. But a key to this is carbon is really common. It's one of the most abundant elements in the universe. And water is really common. It's ubiquitous. Again, we're dealing with oxygen and hydrogen, and these light elements are among the easiest ones to make. So we already know of, or we can extrapolate that there will be tens of billions of wet, rocky, Earth-like planets in the in the Milky Way. So these kind of conditions that we have on Earth at the origin of life 4 billion years ago should be common. And carbon is, if you heat it up, you tend to get CO2 as a gas. That's what's coming out of volcanoes and so on. Carbonate rocks like limestone, if they get subducted into the earth, they come out as carbon dioxide gas. And that's, you could think of it as a Lego brick. It's a small molecule that's not very reactive, so it can accumulate in the atmosphere, but you can snap bits off and add bits on and build molecules of the complexity of DNA from it. So you've got a kind of molecular modeling kit, and the individual bricks, the Lego bricks, are carbon dioxide molecules, and they're, again, common in the atmospheres of exoplanets, but in Mars, in Venus, they're full of CO2, these atmospheres. So now we come back around to your question after that, which is how likely are these processes? And the answer is, well, it's highly likely that we will be starting with carbon dioxide, that it will be abundant and it's very good at its job. And in water, organic molecules form all kinds of interesting structures, membranes that don't dissolve, but form structures. You get free structure from carbon in water, basically. And it's this free structure, you may say, oh, it's a low entropy state, but it's not. It's like a bubble, a soap bubble or something. It wants to form, and because it forms, it releases energy. It's the most stable state it can be in for these molecules in that shape. Just as oil will float on water, it's the most stable state, but you've got some structure there. And that structure releases energy, which in effect is heat, which is increasing the entropy of the universe. So you get free structure. It doesn't. This is following the second law of thermodynamics. Entropy increases as a result of these structures forming. And we're dealing with organic molecules. Hydrogen is split from water in these hydrothermal systems, which are a reaction between rock and water. It's as simple as that. So again, we would expect to find it almost everywhere. And what do you want to do if you want to make organic molecules? Well, CO2 itself is the building block, but organic molecules, if you were to come up with a kind of a very vague formula for life, to a first approximation, it will be CH2O. So you strip out one of the oxygens from carbon dioxide. You'd have a little bit of nitrogen and phosphorus and sulfur and everything else in there as well. But basically, the kind of the hand of cards that you get for carbon-based life is CH2O, and those H2, the two hydrogen atoms, are coming from water or from hydrogen gas or wherever they're coming from. But you need to put hydrogen onto carbon dioxide to make living cells, to make living molecules. And all of these things are abundantly free in hydrothermal systems and would be in any wet, rocky planet. And so we're always going to be faced with the same problem, that we need to get CO2 and hydrogen to react together to form organic molecules, and you need electrical charges on membranes or on inorganic barriers to make that reaction happen. And so it may seem very limited or lacking in imagination, but I think that if we were to find, say, 1,000 different life forms out there in the Milky Way, the majority of them, maybe 990 of them, would turn out to be carbon-based and cellular and not utterly dissimilar to what we see here. Maybe there's 5% of them will be, funky, strange, different things, but just statistics would say this is a good way of doing it based on the most abundant materials in the universe.
So it's basically hydrogen, carbon dioxide, and hydrothermal vents, right? But that all three of them, at least they are crucial. Because then, I mean, of course, people can ask this question that if it's hydrogen and carbon dioxide only, then why not, why there are no life forms in my cola bottle or something,
Well, this kind of chemistry only works in the absence of oxygen, because hydrogen and oxygen really, really want to react together. This is rocket fuel. This is what's powering rockets. So as soon as you've got any oxygen, then the hydrogen will react preferentially with the oxygen to produce water as a waste product. And the CO2 will never get a chance to react. So you'll never form any organic molecules. This chemistry doesn't work if there's any oxygen around. So it's critical at the origin of life 4 billion years ago, the Earth was essentially anoxic. Oxygen came later on with photosynthesis. There would have been perhaps trace amounts of oxygen earlier than that, but next to nothing. And this chemistry only works, so it's never going to happen in your Coca-Cola bottle.
Yeah, so that's interesting that there was no oxygen at the in the early stages of the planet, but then through photosynthesis, as you mentioned, the oxygen came on the planet, and that actually took over the planet. And what is the reason why then now at least all the animals and the species that we see, they are oxygen based. We don't like take CO2 and throw out oxygen, right?
Yes, I mean plants do
Do that.
that. Plants
Yes.
are converting. I mean, they're doing this same basic chemistry. Plants are taking carbon dioxide from the atmosphere, and they're taking hydrogen. They're getting the hydrogen from water. So they're using the power of the sun to split water and take the water is H2O. So they're taking the two H's out of water and cobbling them onto the CO2, leaving the oxygen left behind as a waste product, and that just goes off into the atmosphere. And over time, and it's not a simple process either, but over time collects in the atmosphere. So plants are doing exactly the same chemistry that I was talking about, reacting hydrogen and CO2 to make organic molecules, but they're doing it in a more sophisticated way and taking the hydrogen from water. And animals are more or less reversing that. What we are doing is eating plants. Maybe we're eating animals, but we're the animals have eaten plants, and so we're basically eating the organic matter that came from plants. We're stripping out the hydrogen from it, and if you've got this kind of general structure for organic matter of CH2O, we're breathing out the CO2, the carbon dioxide, and the hydrogen, there's 2H in there, we're feeding it to oxygen. So we're basically powered by the same thing that rockets are powered by. We're burning hydrogen gas in well, not gas, but hydrogen atoms in oxygen, and using that to power everything that we're doing. And the waste product of that is water, which we breathe out. So photosynthesis is CO2 plus H2O gives organic molecules, and respiration is the organic molecules plus O2 gives CO2 and water. So we're more or less exactly the opposite. And there's a tremendous amount of energy in that reaction, which powers animals to run around and chase each other and mate and predator-prey relationships. Everything that we're familiar with animals doing requires a lot of energy, and that energy is coming from really the properties of oxygen, which mean it's stable enough to accumulate in the atmosphere to high levels, 20%, 21%. It's stable enough to do that, but it's reactive enough when you feed it electrons one at a time in respiration to provide all the energy for a really high octane lifestyle.
But in the early stages, most of the work was done by bacteria. The bacteria did it, like it was, I think, mainly cyanobacteria, that they once they were evolved, they could simply trap carbon dioxide and emit oxygen.
Yes, they did, but there's a lot of controversy about the timeline and when exactly it happened and how exactly it happened. It may be that it happened as early as 3 billion years ago. I mean, some people think even earlier than that, but there are what are called whiffs of oxygen, which is to say there are some slightly oxidized minerals from about 3 billion years ago that look as if they may have been oxidized by oxygen in the atmosphere in small amounts. But the evidence is equivocal, and it may be that it wasn't until 2.2 billion years ago. But there's a tipping point. I described respiration and photosynthesis as equal and opposite processes, and there are other processes. For example, the formation of methane by methanogens. Well, methane will react with oxygen to form CO2 and water again, and it won't really leave a trace of itself. So if you've got a balance between photosynthesis producing oxygen and methanogenesis producing methane, and the two react together, then you would not accumulate any oxygen in the atmosphere over the long term. So it's only when something prevents the oxygen being consumed in this way. And the easiest way for that to happen is if organic matter gets buried in the earth, so it's not reoxidized again by respiration and degradation and so on. then oxygen can accumulate. So we don't really see an accumulation of oxygen. We see an initial burst in a time called the Great Oxidation Event around 2.2 billion years ago. And then we go into a long period of stasis after that, which is sometimes called the boring billion. And then we see a bunch of kind of global scale, almost catastrophes around about 700 million years ago. Snowball Earths, where the whole planet froze over right down to sea level at the equator. And then soon after that, oxygen, or around that time, oxygen is beginning to accumulate almost to modern levels. And that's when the Cambrian explosion happens, when we first see very quickly in geological time animals appearing in the fossil record. And it's not that oxygen necessarily drove the emergence of animals so much as you can't really have complex ecosystems with predators and prey and multiple trophic levels in the absence of oxygen, because there's just not enough energy in the system.
So, I mean, we can thank those conditions also to exist, because that's why we, as in like animals, we exist on the planet, right?
Yeah.
And what are your thoughts on the eukaryogenesis? I mean, of course, once we think of the Krebs cycle, which respiration, the process of respiration that you explained, it is mainly done in bacteria, in the sense, and bacteria in our case would be mitochondria, where it's done. So because that is again another step which requires another conception, and we don't know much why or how this would happen, right?
Yes, I mean there's a lot of controversy about all of this, but the bottom line is all recognizably complex life on Earth, and when I say recognizably, I'm talking about things we can see, and animals, plants, fungi. We all have the same type of cell, which is so we're all called eukaryotes, and the word eukaryote means true nucleus. And the nucleus is the compartment in a cell where all the DNA is stored. And bacteria don't have that compartment. They're much smaller, in the order of 10 to 100,000 times smaller by volume on average. And they don't have obvious compartments. They do have some form of compartmentalization. They do have amazingly sophisticated molecular machines. At the level of biochemistry, bacteria in many ways are more complex than eukaryotes are. But in terms of their morphology, their size, and their apparent complexity, they're very simple in comparison. And there's a bit of a mystery that all eukaryotes, plants and animals and so on, if you look at their cells under a microscope, the cells are practically identical to each other. They differ in the proportion of the structures, and they can differ in their overall appearance. But if you look, does this cell have a nucleus? Then a plant cell has a nucleus, and an animal cell has a nucleus, and so does a fungi. And if you look at the structure of the nucleus, you realize, yes, it's got this elastic net around the inside of the nucleus, and it's got the pore structures, and it's the same proteins in the pore structures. And the DNA is coiled in exactly the same way around exactly the same type of proteins. And they divide by mitosis on a spindle of amazing microtubules that pull apart the chromosomes. And we all do sex in an ancestral way, just meaning as egg cells and sperm cells fuse together and then go through this kind of crazy dance of meiosis where the chromosomes line up and then double themselves, and now we've got 4 chromosomes all lined up, and then they crisscross between themselves and then go through 2 rounds of cell division. All of us do that. Plants do that. Amoeba does that, and so on. And in the outside the nucleus, in the cytoplasm, we've got the same membrane systems. We've all got mitochondria. We've all got the same kind of cables and motor proteins that are doing work. So the eukaryotic cells are amazingly similar to each other. And if you kind of try and reconstruct, well, what happened? What came first? How did this level of complexity arise? You're left with a problem from a phylogenetic point of view. So what phylogenetics is doing is comparing gene sequences and saying, okay, well, this branch is here and that branch is there, and we can reconstruct a tree of life. And you look at eukaryotes and you say, okay, so how do these genes branch? And the answer is, well, they all seem to go back and they branch together right at the beginning of the eukaryotic tree. All eukaryotes have all of these genes, and they've all diverged about equally between the different groups. And so you say, okay, well, what about what would an outgroup be? How can we distinguish what happened at the beginning? And the answer is, well, there's bacteria over there, and there's another group called archaea that look a lot like bacteria, but they're biochemically and genetically quite different. And they don't have any of these genes or any of this morphology. And so you're left with I've called it a black hole at the heart of biology. All this complexity in eukaryotic cells, we don't have an easy way of getting at how it arose. And we don't have a clear explanation for why bacteria never developed that level of complexity. There's plenty of ideas around. The problem is that it's very hard to prove these kind of ideas, and so there's lots of squabbling. It's a big, important subject, and different people have a different lens on that question. We're talking about something that happened probably 2 billion years ago, and so it's hard to reconstruct. So, of course, there's arguments. My own take is that it's not about genes. It's not about information. You get this as a recurring theme from me. I don't think genes are the only thing in biology. I'm not trying to, I've been talking about gene trees and comparing these structures. Genes are incredibly important, but they're not the only thing in biology. So if you think about bacteria, they have a different way of organizing genetic information. What they tend to do, you think of an E. coli cell, it might have 3, 4, 5,000 genes. E. coli can differ by 2,000 genes between two different cells. They can differ in their gene content a lot. So let's say 4,000 genes. The reason they can differ a lot is that this is a single cell has that many genes, but if you look at the different strains of E. coli, they can differ by 50% of their genome. This is more variation than in the entire vertebrate tree. How do they get to be like that? Well, they have a big metagenome, which is to say genes out there in the environment in different populations of E. coli that get swapped around, sometimes on a big scale, by what's called lateral gene transfer. And I like to think of it as kind of loose change, where it passes around from one individual to another one. They're not vertically inherited in the same way. They're just passed around as a kind of network of how interactions between individuals, like passing money around. So there may be 30,000 genes in this metagenome. And the way that most bacteria seem to work is they prefer to keep their own genome as small and as streamlined as they can, because that allows them to grow faster. But if they run into problems that they can't grow, then the easiest way out is, okay, I know I've got a problem, so I'll pick up some genes from out there in the environment and I'll weld them into myself and I'll see if I work better then. And that's the kind of the bacterial philosophy of life, you might say. What we can say about bacteria is that over 4 billion years of evolution, they haven't really changed. We can see fossils that look a lot like bacteria from 4 billion years ago. And we can see definite traces from 3 billion years ago that are very clearly bacteria. And we can see in the minerals signs of the metabolism that the different types of cell had, for example, including photosynthesis. And so we can reconstruct a bacterial history of the Earth. And they're still out there. They're still doing the same things, and they've never become large and complex. You never see a flea made from bacterial cells. So why not? What's the problem? The reason I don't think it's a problem with genes and information is that they have experimented with more genes and more information and more sequence space than eukaryotes ever did. They've done it on a much bigger scale. They have practically infinite populations. They've had practically infinite times. They've got huge metagenomes, bigger than the human genome in E. coli. And they've done all this shuffling and rearrangement and everything you could imagine from an information point of view. And yet they got stuck in a rut at a bacterial level of complexity. So what's different about eukaryotes? Well, we have mitochondria. And mitochondria were bacteria, and bacteria got inside another cell, and there's lots of arguments about when or how, but in effect, what we end up with is multibacterial power without the overheads of having complete bacterial genomes in every single power pack. And what that allowed us to do is swell up the nuclear genome so we can have 30,000 genes, 20,000 genes in a single cell. And that means that we can grow from a single egg cell, a fertilized egg cell, and all our cells are genetically identical, but the cells in the brain, or neurons or astrocytes and glial cells in the brain, express very different genes. They switch those ones on, they switch those ones off, and in the kidneys it's different, and in the liver it's different, and in muscles it's different. And so we have the power to store all of these genes and then switch on different ones, and that means we don't have a kind of conflict between the cells in an individual, because we're all genetically, as an individual, a clone. And that seems to work in evolutionary terms. Whereas if you bring genetically different cells together, what you get is something like a bacterial biofilm. And these cells they kind of get along, but they're always fighting in one way or another. They're always cheating. They're always trying to short circuit the system. And so you get a level of complexity which is equivalent to a biofilm, which is to say, you would recognize it as a sticky patch of cells, but not as a complex organism, really.
So one of the strongest evidence that we have that mitochondria are literally bacteria is the fact that both of them, they could do this process of respiration, and they use like similar processes at the
It's
end.
basically exactly the same machinery. Is more pared down in the bacteria, but our own mitochondria still has DNA. That DNA is basically bacterial DNA. They have ribosomes, the protein building machines mitochondria have got their own type of ribosome, which is different from the one in the cytosol. And it builds respiratory machinery for burning food in oxygen, which is exactly the same structure of all the proteins that we see. They're more complex. There's more parts to them in eukaryotes, but the core of the machine, the working part of the machine, is exactly the same, superimposable in terms of molecular structure and the mechanics of how they operate. So there's no serious question. There's always people out there who say, "Hang on a minute, I'm not sure I believe this." But to 999 out of 1,000 people working in this kind of field, there is no question that the mitochondria derived from bacteria, and that their genomes were whittled down from a standard bacterial sized genome of 3,000 or 4,000 genes down to we're left with 38 genes in our own mitochondria, of which some of them are involved in building the proteins, but a few of them are actually encoding the core subunits of the proteins that are responsible for respiration. So this, to my mind, is the most important step change in all of evolution, that we go, in a really trivial sense, from a population of cells that is limited in its topology, that we've got an electrical charge on the membrane surrounding the cells, to a population of cells within cells where the electrical charge has been moved inside. So the bacteria got inside, and now we have an unstable system that sheds genes from the mitochondria and accumulates them in the nucleus. So now we have a genomic asymmetry with a swollen nuclear genome and tiny mitochondrial genomes. All these electrical charges are retained inside the cell, freeing up the cell membrane to do other things like phagocytosis, eating other cells. And the genes that are required to maintain this powerful electrical charge in a stable way have always been retained as mitochondrial DNA. We all have mitochondrial DNA. We inherit it from our mothers only. It's uniparental inheritance, which is to say, your mitochondria, my mitochondria are a dead end. If we have kids, I have kids, the mum's mitochondrial DNA that they have. So I could be a champion athlete with wonderful mitochondria. I'm not, but if I was, then it doesn't matter. For my kids, they get their mum's mitochondrial DNA. She perhaps is a champion athlete, and then they will be too, because they've got these champion mitochondria, if you like. They're inherited clonally from the mother only.
Yeah, we'll definitely talk about this point, but before that, let's talk a little bit about membranes and potential or the electrical charge that you mentioned. So, I mean, of course, all the cells that we have, this is like one physical barrier that we have between life and or animate and inanimate matter. But it's not like really solid barrier. It is a barrier which is fluidic, which can exchange information being signals or chemicals with
Yeah,
the
right.
environment, right? Um, but apart from that, especially in the eukaryotic cells, as you mentioned, the other organelles, they do have membranes, including mitochondria, but then there are also ER and other
Yes. Yeah.
organelles
Yeah.
in the cell. So how the electrical potential is created on the membrane?
So this was one of the most important discoveries of the 20th century, from going back to Peter Mitchell and Jennifer Moyle, who did all the experiments. So Mitchell had these kind of crazy ideas, and Jennifer Moyle made them real, I would say. And a bunch of other people doing experiments on it. But the idea, I suppose, going back to that time, is that a cell is a bag of chemicals, and the membrane sits around the cell and closes it, and it's pretty much a semi-inert barrier to the environment outside. And Mitchell had been thinking about bacteria in this way, and realized, well, no, it can't be semi-inert, it has to be active, because there's a difference between the inside of a bacterial cell and the outside world. The inside accumulates all kinds of things, and all kinds of wastes are pumped out over there. And so you need to be able to tell the difference between the molecules that you want to have inside and the molecules that you want to kick out. And that requires energy, and it requires some kind of discernment. You need to be able to recognize this molecule from that one, take this one and pump it over there, which costs energy. So that's, in a really trivial way, where Mitchell was coming from. It was, how do bacteria keep their insides different to their outside? And he was thinking about pH and acidity and protons, and this is one of the key points about how a cell maintains itself. If you go into an acidic environment and you allow all these protons to come rushing in, then it's going to disfigure all your proteins, and you're going to die almost certainly. So we need to recognize protons and pump them out. But he realized there was, at the time, there was an appreciation that the membrane was somehow important in respiration, and it wasn't really quite appreciated how, or if it really was, or quite what was going on. And he conceptualized that protons were being pumped across a membrane somehow, that the energy of respiration, taking hydrogen from food, passing it to oxygen, this releases energy, that energy is stored not in the chemistry itself directly, but in a charge across the membrane. And this was a radical way of thinking about things. And it took a long time before it was accepted. But effectively, when you strip hydrogen out of food, what you're stripping out is electrons and protons. The hydrogen atom is one proton, one electron. We have more or less a wire within the membrane, which is all this respiratory machinery that I was talking about. Inside it all, there are these little clusters of iron and sulfur, the same things that we find in the vents at the origin of life, what we call redox centers, but basically an electron can hop from one of these to another one. You could think of that in lily pads in a lake or something. There's just like frogs hopping from one to the next, and electrons hop down this respiratory chain, and they arrive at oxygen. And each time they land on a redox center, it shifts the balance within a protein, and that provides the energy to pump a proton out. And so what we end up with, we've got the electrons and the protons. The electrons go straight to oxygen. The protons cross the membrane. So now we have a load of protons on one side and fewer protons on the other side. And protons are positively charged. So now we have an electrical charge on this membrane, and the protons coming through the ATP synthase are driving this motor, driving the synthesis of the energy currency ATP. But really, the ATP is a stable energy currency. You can think of it as coins, but it's the thing which is actually powering life is not the ATP itself so much as the charge on the membrane. That's where all the work is being done. And if we lose that charge on the membrane, that's when we die. It doesn't matter how much ATP we have. If we lose that charge, that's the end. So we need to, respiration is continually regenerating that charge. So it's a marvelous mechanism, and it's universally conserved across all of life. that effectively we're splitting protons and electrons. The current of electrons flowing to oxygen is powering extrusion of protons. So we've got a positive charge with a difference in proton concentration between the outside and the inside. And that is how life is structured. And it says membranes are centrally important. And if we're talking about bacteria, that's the plasma membrane surrounding the cell. And it is an energized membrane with an electrical charge on it, and that charge is powering work. And that is the fundamental structure of a living cell.
This is just fascinating. I mean, like in my case, I mean, since I've gone through your book, I read it. So the book is Transformer: Chemistry of Life and Death. Fascinating book, and all the stories that you've mentioned about discoveries, it was again amazing. And Peter Mitchell, who got the Nobel Prize, but again, it was it was work of two, but only
Yes,
the
and this
guy
is
in another story got the
yes, and I hope that if people do read this book, that they'll take those stories on board. I mean,
yeah.
I had to dig around a little bit with this story because lots of people mention Jennifer Moyle's name, but she's often dismissed as if she was a technician who was told what to do. Um, and there's something wrong. Something doesn't smell quite right to me. And I've talked to various people who knew her, and they almost have reinforced that view. But when you read the papers, there's clearly a difference between the papers that Mitchell wrote by himself as a single author. And there's no doubt that he was an imaginative genius. He came up with some amazing ideas. But you read the papers that he co-wrote with Jennifer Moyle, and you realize that, well, she had a big input on this. She was not a technician. She was, and you read about her own background, and she was a brilliant woman. There's no question about that. I don't think she got anything like the credit that she deserved. And the papers that I like best are the ones where she was a co-author and she was bringing a kind of analytical, scientific precision, of experimental precision to the questions that I think Mitchell perhaps lacked a little bit himself. This would be a scurrilous view to a lot of the people who worked on the chemiosmotic hypothesis. They'd probably disagree with me violently, but I am trying to reconstruct what I think happened there. And I think she was much more important than she's given credit for. And I think she deserved the Nobel Prize, or sharing the Nobel Prize with Peter Mitchell.
Yeah, it's a great job that you're doing, and recollecting those stories. It's I hope that we won't do the same mistakes in 21st century at least.
I really hope so
So
too, yes.
yeah, and thinking of such a fundamental process, it's just fascinating. And so in my conversation with Randy Sheckman, Nobel
Mm-hmm.
laureate for
Yeah.
2013, um, his work on autophagy. So he said that about membranes, that the membranes are also not linked with information, uh, in a way that all the membranes, they are derived from the previous existing membranes. It's not like there are
Yes.
genes or
So
something.
there,
So
I mean, maybe what you mean there is they're not linked with genetic information, with DNA.
indeed, indeed, yeah,
They have their own information, and
yeah,
it's called membrane heredity sometimes. And
yeah.
yes, there is nothing in the DNA of the cell that says go make a membrane over there. It's coming from the membrane that's already over there. That's where the structure of the cell is coming from. He's absolutely, well, of course he is, but he's absolutely
Yeah,
right. Yes.
yeah. I mean, this was fascinating for me because most of the people who are working on this replication first kind of view, the focusing on information, this is what they say, that it's the membrane formation is quite spontaneous. That you just put on some lipids, you get some vesicles, and it's easy that it can get trapped and stuff. But on the other hand, there is no link that we have so far when it comes to membranes and these kind of primitive processes that
Well,
they will.
I have tried to, with colleagues at UCL, tried to um, figure out that link, because I keep talking about membranes powering work, and the simplest proteins that sit in these membranes are powering CO2 fixation, which is to say they're powering growth. And so you need a membrane, and it needs to be an electrically charged membrane, and it needs to use the difference between the outside and the inside to drive CO2 fixation inside, in other words, autotrophic growth. So how would it do that? Well, we imagined and modeled it computationally, but we've done experiments since which show that the model is actually closer to the truth than I would have imagined. What we imagined was that if this is true, and that you've got the difference between the inside and the outside can drive CO2 fixation to make organic molecules, we thought, based on thermodynamics, some of these molecules, the majority would be amino acids. And a proportion of those amino acids would tend to interact with iron and sulfide minerals, growing crystals of these things. And we know that these can drive CO2 fixation. That's been shown already as an inorganic barrier. And we know that proteins like ferredoxin, which are really important in CO2 fixation, contain little clusters of iron sulfur mineral, in effect, at their heart. And so we thought, well, if we're forming organic molecules and they interact with iron and sulfur to produce, let's say, smaller crystals, and those smaller crystals can go to the membrane and they can help drive CO2 fixation and therefore growth, then physically what you're inheriting is the membrane itself, and you're inheriting the crystals of iron sulfur crystals collated by organic molecules sitting in the membrane. So it's a physical form of heredity. It's basically a positive feedback mechanism. The more there are there in the membrane, the more they can fix CO2 and make organic molecules. So the more you will interfere with the growth of iron sulfur minerals, and the more they'll end up in the membrane. So there's a very simple positive feedback circuit there. Which is driven by a form of membrane heredity. So we started doing experiments to test whether any of this was even remotely true. And we were amazed to find that if we mix the amino acid cysteine, which is the amino acid that normally would bind to these iron sulfur clusters in proteins today, with ferric chloride, so just a solution of oxidized iron in water, and sodium sulfide, and we mix them together in the absence of any oxygen, then we spontaneously form what are called 4Fe, 4S clusters. So these are exactly the same structures that we find in ferredoxin in all these proteins that are driving CO2 fixation. It's not that we get slightly smaller crystals and slightly smaller as I was expecting. We just go from a large crystal to a tiny 4Fe, 4S cluster in one step, just like that. It's amazing. And we showed that they really are these, that they really are redox active, which is to say they really can transfer electrons potentially onto CO2. It's better than we thought it would be. And the long and short of all of that is this idea of early membrane heredity is, well, let's just call it not stupid. It is partly real. Whether it can do all the work that I'm attributing to it, that's not answered yet fully. But a few years worth of experiments already shows that there's more reality to it than I would have dared to imagine before when we started doing the modeling. You kind of think, well, this is in silico. This is a computer model. It's not reality. It makes some predictions. Are those predictions possibly true? And then you do the experiments. You think, wow, actually, some of it really is true.
Yeah, that's completely a bliss. Once we get that, our predictions they go or they are correct in a way. So so here so basically what we've been talking about so far is that, of course, the information is important for life, but then there is something going beyond information. And according to you, that's that's fixing hydrogen and carbon in the living circle, carbon dioxide. Mm. And this happens in the present life forms through something called Krebs cycle, but that's like just that's basically production of hydrogen and carbon dioxide. But then there is something called reverse Krebs cycle,
Yes.
which actually uses hydrogen and carbon dioxide and gives us products, right?
So most people, if they've heard of the Krebs cycle at all, or it's taught in schools a little bit, anybody who does biology or biochemistry or medicine or anything at university will be familiar with it. And what they'll be familiar with it as is everybody is taught it and they forget it immediately because it's a complicated circuit of reactions that seems to be switching bits of carbon and bits of hydrogen and bits of oxygen around the place, and you kind of get very cross. Well, what's it doing all of this for? And the bottom line is what it's really doing is pulling hydrogen out of these carboxylic acids. and is passing the hydrogen into the respiration, as I was talking about before, feeding them to oxygen. And so the Krebs cycle is basically pulling CO2 and hydrogen out of these organic molecules, the Krebs cycle intermediates, carboxylic acids. Now, it turned out in 1966, and it took a long time for any of this to be accepted, I mean, more than 20 years, practically a quarter of a century, that some bacteria do exactly the opposite of that. Not quite exactly the opposite, but roughly the opposite of that. Instead of stripping out hydrogen and CO2 and burning the hydrogen to generate energy, they're putting in CO2 and hydrogen and a bit of energy to make the Krebs cycle intermediates. So they're turning gases, CO2 and hydrogen, into the building blocks of life. And they're doing that in exactly the same series of steps that you see in the Krebs cycle itself, with a few extra ones added in. And when you think about that chemistry, the chemistry of the Krebs cycle as we know it, you think, I mean, there is some logic to it, but you sit there and you think, well, why do you start with glucose and then break it down to pyruvate and then turn pyruvate into all of these? Why do we do all of this stuff? And there's some erudite answers to it, but they're not, they don't really grab you. That's why everybody forgets the chemistry of the Krebs cycle again, because it's so unmemorable. You're just left thinking, well, why is it like that? It's madness. And I have to learn it for an exam, and I really don't like it. But then you look at, you think about the reverse Krebs cycle, and we're taking gases, the same gases that you find in any hydrothermal vent on any wet, rocky planet, and they're being reacted together. And I say it requires some energy. It requires this electrical charge on the membranes or the barriers to react hydrogen and CO2 to make the key building blocks of life. You react hydrogen and CO2, you get carboxylic acids. That's what you get. And when you draw it out, you can see this is the chemistry you are going to get. And then you realize that the structure of metabolism as we know it, if you want to make the building blocks of DNA, the letters, the nucleotides in DNA, well, we start from amino acids and sugars. That's the starting point in modern metabolism to make the nucleotides. So where do the amino acids and the sugars come from? Well, they come from the Krebs cycle. They come from the Krebs cycle intermediates. You just add a bit of nitrogen to the Krebs cycle intermediates, or some of them, and you get the amino acids that are used for making nucleotides. And to make sugars, I was just talked about breaking it down to pyruvate. Well, pyruvate is part of the reverse Krebs cycle, and that's the basis for making ribose, for example, for RNA, or deoxyribose for DNA. So it's right, metabolism is structured around this. And when you think about metabolism starting with the gases, hydrogen and CO2, then all of it makes just beautiful, perfect sense. And instead of you thinking, why on earth is it this fiddly system with it, it suddenly just smiles back at you as well, practically inevitable chemistry. And it's quite exciting. It's exciting just from the point of view of, how does life start? How does an inorganic, sterile, if you like, wet, rocky planet come alive? And it's just got gases. Well, those gases are reacting together to produce these molecules. And a lot of this chemistry is now being done in the lab. But the other reason it's really important for us is that when we think about our own Krebs cycle, we realize, well, it's doing two jobs. It's doing the job that everybody attributes to it in the textbooks, which is to say it's part of energy generation in cells. We're stripping the hydrogen out and feeding it to oxygen and so on. But these Krebs cycle intermediates are still used today by us, by all animals, as the basic building blocks for making amino acids and for making nucleotides and the rest of it. So we're doing two different jobs with the Krebs cycle intermediates. We're doing the energy generation side, but we're also doing the biosynthesis side. We're making the building blocks of cells from the Krebs cycle still today. And it's complex, and it's often put aside in a separate chapter in the textbooks. We've known about all of this since Krebs himself. It's not new information. But what's really changed is cancer over the last 10, 15 years or so. We've realized that from around about 15 years ago, it was first discovered that there are mutations in the enzymes encoding Krebs cycle enzymes, in the genes that encode Krebs cycle enzymes, that can cause cancer. And so people started thinking about flux through the Krebs cycle again, as potentially carcinogenic. What happens if you've got a mutation here? Well, this intermediate accumulates and leaks out of the mitochondria and switches off those genes and switches on those genes, and suddenly we've got a cancerous phenotype here. So why would it be like that? Well, then you think, well, what cancer cells want to do is grow. They want to make copies of themselves. What do they need to do that? Yes, they need some ATP, but they don't need that much ATP to make new proteins, to make new DNA, to make new membranes, everything you need as a cell. ATP is part of what you need, but not the biggest part. You need the carbon skeletons, you need the Krebs cycle intermediates, you need to make more DNA, twice as much DNA as you had before. You need to make new proteins, you need to make new membranes. So that requires the carbon skeletons, but it also requires what I call the reducing equivalents. You need hydrogen in the form of NADPH today. But effectively, you need to be running the Krebs cycle in reverse, pretty much like the way that bacteria in deep sea hydrothermal vents have been doing it for 3 billion years or something. So you begin to look at the structure of metabolism with this strain on the Krebs cycle in mind, and the way that cancer cells are stripping these intermediates to power their growth. You see it in a very different light. And when you realize that this is the same chemistry that's been working in life over billions of years, so why is it that we are more prone to getting cancer as we get older, and we begin to get damage to respiration, and we slow down a little bit? Well, the Krebs cycle begins to shift its direction, and its use is beginning to change. We begin to put on weight, we begin to become more at risk of cancer, and so on. So it's a shift in the metabolic potential of cells as we get older. And so all of this material about the origin of life and how ancient bacteria work to fix CO2 through photosynthesis suddenly is relevant to why we get cancer. And the book is about putting these two halves together.
And what about recycling of mitochondria? Because it's not like, first of all, it's not like we have only one mitochondria. There are like thousands of them. And then these mitochondria also undergo kind of cell death in a way. Because I also did one conversation with a doctor, Dr. Robert Lustig, who wrote a book on metabolical is title of the book. And he talks about one of the aspect is why we gain weight, etc., is this mitochondria dysfunction, in a way that our cells, they tend to accumulate a lot of bad mitochondria, which should be recycled. And till the time we don't take a shock or something, or give a shock to our body, through exercise, fasting, or maintaining our food intake, etc., it doesn't recycle those mitochondria. It just preserves them somehow. So what about what's that factor?
Yeah, so there's a resource use question here. And it goes back a long way in biology about, in effect, we know that you can that an organism has a choice with the resources it's got of investing more in sexual maturation, or finding a partner, or producing offspring, or whatever it may be. I'm not thinking about humans so much as just life in general, animals, and so on. But it's a kind of a general pattern in biology. Or you can invest more resources in staying alive for longer, which takes away from the resources you're investing in sex and puts more into staying alive, into a stress response, for example, into dealing with an environment that's not a happy environment to be in, if you like. And calorie restriction has been known for a long time to be a way of extending lifespan. It's equivocal whether it really works in humans, but it certainly works a little bit in some rhesus monkeys, for example, and it works very well in things like mice. So the more complex we get and larger we get, the harder it is to make it work. But one of the things that is noticed with calorie restriction is it tends to suppress sexuality and the number of offspring that you're going to have. But there are various other ways of doing it. It's not always a strict relationship that the more investment you put into longevity, the less you put into sex, or vice versa. But there is at the level of the Krebs cycle and flux through the Krebs cycle, there is this question: do I want to be investing in growth, which is to say sexual maturation? So I'm using the Krebs cycle primarily for the intermediates to power my growth and to bulk up and to make proteins and so on. Or do I want to use it primarily for energy generation and doing work? And that work includes autophagy and the breakdown of cells and the breakdown of mitochondria and so on. So how do I want as a species, as an organism, a population of organisms, how long am I likely to live anyway? If you're a house mouse or something, you're small, you've got a fast metabolic rate, you're vulnerable to predators. If you make an investment decision that I'm not going to have any offspring for at least the first 5 years, well, the chances of you living to 5 years old is virtually zero. So it's a bad decision. It'd be much better to invest much more in reproduction earlier on. So this is a standard kind of evolutionary equation, but it boils down at the level of the Krebs cycle to a decision on, is this flux going to go this way or that way, or how much is going to come in here and out there? How much, there are real structural constraints on metabolism that are saying you can't do both things at once all the time so well. There's only certain ways that you can do it at the level of the wiring of metabolism. And then the other thing that I think is very easy to overlook, we all know it, we're all taught it, but we easily forget it because we can't see it, is the number of metabolic reactions going on at any one moment. We're talking between 1 billion and 20 billion, maybe 30 billion reactions every second. It's an extraordinary number. Second after second, as I'm saying this, 100 billion reactions just happened in one of my cells, and I've got 40 trillion cells, and each one of those just did 100 trillion reactions in the time I was saying that. It's incomprehensible. So how much investment do you put into getting all of those reactions exactly right so they always work? And what I'm falling back on here really is an old-fashioned and slightly outdated thermodynamic idea that stuff goes wrong over time. And this is a tremendous amount of stuff that's happening. And it's amazing that we live as long as we do, really. But we accumulate damage over time. And I think this is one of the themes from aging research. A lot of people working on diseases of aging and so on will recognize the damaged proteins, accumulation of damage, and they should be turned over. They can be turned over through processes like autophagy and so on. So what's going wrong? Why are they not in the end turned over? Well, there are these tipping points that I've been talking about. As we accumulate damage in respiration, then the flux through the Krebs cycle and which genes are switched on and which genes are switched off, and the activation state of the cell, if you like, where cells are flipping to something approximating to a senescent state as we get older. And a lot of it is linked to flux through the Krebs cycle and all the signaling that comes from that. And that signaling is more or less saying, don't break down those broken mitochondria, don't break down this kind of damaged endoplasmic reticulum. Let's just grow instead. It's a kind of the signal is wonky. The signal is wonky because damage has accumulated. Damage accumulates because of all of this metabolism that's going on so fast that it's practically impossible to stop it accumulating. And so we end up with a signaling system that's reflecting some level of damage in the system, and which is then switching on the wrong set of genes. It's doing that because the flux through the Krebs cycle is shifting in the direction that says grow rather than the direction that says repair. And that's about, are you using the Krebs cycle for energy generation, which you can equate with repair, or for growth, for using the intermediates for powering the growth of cells, which to a first approximation, you can say, forget about repair, just make more of me. And that's why cancer is, is something like 100 times more likely by the time you've got into your 50s compared to when you're in your 20s. And the risk goes up, it doubles every 10 years or something. My risk will have doubled by the time I'm 60 something. It's to do with age. It's not to do with the rate of accumulation of mutations. It's to do with the way that the system is working and the signaling is working. And that in the end is down to metabolism and not genes.
I mean, definitely we can't stop that process, but can we slow it down? Are you working on aging also, or do you have?
A little bit. It's not really my main interest. I'm working with people who on flies, for example, and the way that mitochondria work in Drosophila, and that touches on aging. And they're very, very interesting questions. I don't. I'm not really an expert on that by any means, but being an expert is not always the best thing to be. I think sometimes blundering around in the undergrowth of the kind of thickets of how do things relate to each other, um, you're ignorant about a lot, and you're wrong about a lot, but you see shapes and patterns that maybe an expert might have overlooked because they're not blundering around in the undergrowth wondering how things fit together.
Yeah, I mean, I remember now again the story of Peter Mitchell, what you wrote in the book. It was something similar that he was more focused on philosophy of bacteria or something that.
Yes.
So that's yeah. So it seems like
I mean,
similar examples.
it's a common pattern, really, that a lot of breakthroughs in the history of science have been made by people who came from outside the field, and the in a way, the less you know, the more likely to ask the childlike questions you are. Why is it like this? Um, and just framing those questions is part of the way to answer them. And the more you know about something, the harder it is to see with fresh eyes. The harder it is to come up with something really creative and original and unusual and different. And that goes, I think, it's a human thing. It goes for all of us. The more you know about something, you may as well say the more jaded we become. And this is, there's a practical side to it. When I have new PhD students starting, they tend to be encouraged to go and do a literature review, learn everything you can know about this subject, and then work out what you're going to do that's new. And I think it's completely the wrong way to do it, because it's so off-putting. It's so overwhelming. There's so much information out there. You read all of this stuff and you think, well, everything that's possible to know is already known, and I am very inadequate, and I have no contribution to make. I think I just will forget my PhD and go and sell ice cream instead or something. I don't know. I did have one student who went and became a circus performer, and I really, really kind of think, what a brave decision. But it's kind of symbolic in a way. And the problem is people get overwhelmed. Now, if you are talking about the blundering around in the undergrowth and not knowing too much, asking naive questions, that leads to, how do you test this naive question? What kind of an experiment can I do here? What kind of kit do I have access to that I can do this naive experiment? And so you end up doing things, and then you look at the literature and you think, oh, this was all done 20 years ago by someone, but they didn't do this and I did that, and the results I get are different to theirs. And then suddenly you look at the literature in a different way and you realize that, well, no, everything hasn't been done already, and people make decisions about what to do. And when you write a paper, you write it in a way that pretends to be foolproof so you can get the editors to accept it. And it's not. It's riddled with holes. Every paper is riddled with holes. Our knowledge is very fallible, and it's a much more productive way not to know too much and to go on asking the naive questions. And I think that's why so many breakthroughs are made by people from outside the field who come in and wander around without knowing very much, asking the most interesting questions.
Yeah, I mean, it's not a naive question, but I think one of the great statements that I read in your book, to quote you, I suspect that 21st century biology will be the biology of fields, which is really interesting. I mean, so of course you talk about electric potential on the membranes, and that's being the electrical fields in a way. And I had a conversation with Michael Levin, who talks about
Did you? Yes. Okay.
bioelectricity,
Yes.
and you've been talking about his work as well. So, what are the other fields that you think? It's mainly the electrical fields, electromagnetic fields,
I'm
or are
thinking
we going beyond?
electromagnetic fields. How real this is, I honestly don't know. But I think it probably is. So, I mean, we know, the EEG, the electroencephalogram in the brain, or the ECG in the heart and so on, we know that it reflects electrical activity and changes in voltage. And these are on quite a large scale. And so they involve networks of cells, hundreds, thousands of cells involved in a single electrical change that we could measure with a probe from outside. But we don't have a very clear idea of exactly which bits of all these hundreds of cells that are cooperating together to produce some kind of change in voltage, change in fields that we can measure. Which bits of the cells are doing that? And I would not have thought about this really at all, except for some, I think, beautiful work from Luca Turin, who won't agree with everything I'm saying about this, but let me just say what he says first. He is interested in the mechanism of general anesthetics, volatile anesthetics, and he has shown that, for example, gases like xenon, which is an inert gas, act as a general anesthetic. So it's not because of their chemistry, really, because they don't. It's an inert gas. They don't have any chemistry. And it's not because of their shape that they're fitting into some receptor. And actually, this has been a problem for a long time in the field of general anesthesia. All these general anesthetics have completely different shapes and structures. And there's no obvious pattern to it, except they tend to be lipid soluble and they tend to accumulate in membranes. And so Luca Turin had shown that general anesthetics interfere with the transfer of electrons to oxygen. Now, that doesn't prove that it's a direct causal relationship. It could be that they're interfering with the transfer of electrons to oxygen, so respiration and the charge on the membranes, and obviously that's going to affect the energy available to the central nervous system, and so any process that generates consciousness is going to be affected by that. So this is not a causal proof, but what it's saying suddenly is that here's a gas that's not doing anything else. It's xenon, and it's directly interfering with the transfer of electrons to oxygen. And we've already established that there are these electrical patterns in the EEG and so on, which are reflecting our conscious state of mind in one way or another. It's different when we're asleep. It's different if we're under a general anesthetic and so on. And the other thing which I would say has changed in recent years is the realization that these are not epiphenomena, which is to say, this is not a side effect of the real activity of neurons, that they can't do anything themselves. They're just a kind of an epiphenomenon. Because if you cut an axon and you separate the two parts by about 40 up to 40 micrometers, then an action potential which is coming down here can just hop over that gap as if it didn't exist. And it's not because there's neurotransmitters crossing the gap or anything. They're not that fast. It's as if that gap doesn't exist at all. It's an electrical hop. It's done by electrical fields. And what that says is that electrical fields are not unable to do anything. They're not unable to make work happen. It can power the transmission of an electrical potential down a neuron just through the field. So fields will do work, and they're interfered with through general anesthetics, and general anesthetics interfere with the transfer of electrons to oxygen. And mitochondria are they're inside cells, inside neurons. But a neuron, the membrane on a neuron, the plasma membrane of an axon, the membrane potential has been measured over decades. It's in the order of 60, maybe 80 millivolts between the inside and the outside. Whereas the mitochondrial membranes are potentially twice that. They may be 150 up to 200 millivolts. Now, that's problematic. That's a problematic statement because nobody has ever measured it using a microelectrode in the way that people have with the neuron. We measure it using dyes instead, which accumulate according to charge. But there's all kinds of problems with the use of these dyes, and we don't really know what they're telling us, and we don't really know how to calibrate them. And mitochondria are not at all this kind of open, semi-open system that they're often drawn as in a textbook. And if you insert a microelectrode into a mitochondria, and the people have done this, Tedeschi was doing this in the 90s or earlier, and they've never measured anything more than about 10 millivolts, 10, 20 millivolts at the most, calling into question the whole of chemiosmotic coupling and Peter Mitchell's hypothesis and so on. So are they right? They're good scientists. They're doing serious work. I believe the measurements that they've made. So what have they measured? Well, I think what they've measured is the difference between the mitochondrial matrix, which is the main part of the mitochondria where you would insert your and they would inject dyes through these electrodes, and they go into the matrix, and the cytosol, which is to say outside the mitochondria. But where all these protons are being pumped is into what's called the Christie space, which is not continuous with the rest of the, turns out now, is not continuous with the rest of the inner mitochondrial membrane, but it's a closed space, and it's a very narrow space, and protons are being pumped into there, which means that the charge on those membranes is where all that charge really is. And it's hard to measure, and no one has ever succeeded in measuring it unequivocally. But it almost certainly is in the region of 180 millivolts, and it's in that space. Now, the other things that have changed is that this closed Christie, where all the protons are going, at the end of it is bent by dimers of the ATP synthase. So they sit at the end there and they bend the membrane. And all the rest of the pumps, the respiratory complexes that are pumping protons into this space, they're on the side walls. So we now have a circuit of protons which are going up to the ATPase dimers at the top and then back down and being pumped through and up. And so we've got a current of protons going on. We also have a current of electrons going to oxygen, but I suspect it's the protons that are doing the work here. And we have parallel Christie, potentially, each one with an oscillating current of protons, which could be in phase and could generate electromagnetic fields. This is where Luca Turin begins to doubt whether or not these fields are really doing any work. He may be right. I don't know. I mean, it's practically impossible to say at the moment, because all of what I'm telling you is quite new, quite difficult to measure, and right at the edge of what would be respectable science. You mentioned Michael Levin. He's been showing over the last 10 years or so that development of many animals, he's shown it beautifully on flatworms, planaria, but it's more broad than that, is determined by a language which we hardly know at the moment of electrical fields, which effectively tell cells what to develop into and how long to keep growing and replicating and so on. And he's done amazing work. I mean, gobsmackingly beautiful work, showing that these fields are controlling development. So we really do know now that electrical fields or electromagnetic fields are influencing the environment in which cells are behaving, how they grow, when they stop growing. Now, what if these fields are coming from mitochondria and are being amplified by mitochondria? You may say, well, why even bother thinking that way? Why single out mitochondria? I've said it's because there's many more membranes. They're parallel very often. They can, in principle, generate these fields which could operate over distances, perhaps as far as the plasma membrane, and interfere with the charges on the membrane. But why? Why the mitochondria? Well, remember, they were bacteria once. And when they were bacteria, the membrane that we're talking about, the charged membrane, was the plasma membrane facing the outside world. And so it's how a bacteria is behaving depends on the Krebs cycle again. It depends on, is hydrogen being fed into the Krebs cycle, or have we run out of supplies? Are we starving? Is there enough oxygen to burn this stuff in? Do I need to grow? Are my requirements for growth okay? All of these, I said, 1 billion reactions a second of metabolism happening inside a single bacterial cell. All of this stuff is happening across the cell. How does one end of the cell know what the other end of the cell is doing? It's not, we're talking about picoseconds here. We're talking about, picosecond by picosecond by picosecond. Something over there knows what's happening over here. How? if we're talking in the language of fields, then it begins to make sense. All of this metabolism in the end is structured through the Krebs cycle to charge a membrane, and the charge on that membrane is going to tell you in effect how I am doing in relation to the environment. Have I got enough substrate? Have I got enough oxygen? Have I got enough food? Have I got is my respiratory chain functional? Do I have a good charge that's going to allow me to do the next thing I want to do, or have I just collapsed it a little bit and need to move over there? So it integrates the cell as a single unit, and it tells you how you're doing right now in relation to the outside world. And that seems to me to be what you would want the simplest form of consciousness to do. So I'm not saying it's an awareness, or at least not an awareness that you're aware so much as an integrated circuit at the level of living cells, bacteria included, which tells you how you're doing, whether you're comfortable or uncomfortable. If you look at the work of Mark Solms and Carl Friston, for example, they talk about Friston free energy, and they're looking at it in terms of homeostasis. And at the level of a single cell, what I'm talking about really is homeostasis. How do you know if you're in the right state or if you're in an uncomfortable state? Well, you know from the charge on the membrane and from the fields that are being generated by that membrane. Am I in a homeostatically low free energy state and comfortable, or am I in a kind of a uncomfortable high energy state that I need to move over there or do something different? And so why the electrical charges from mitochondria? Because they were bacteria once, and because these are what was telling bacteria how to respond to their world. So what I'm suggesting the central nervous system has done is effectively use this as a unit of currency for what a feeling is, for telling you how you're doing in relation to the world, and then a complex central nervous system is enormously amplified, but we still don't know, can't agree what feelings are. We know how neural networks might compute things, and Roger Penrose for a long time has said consciousness is not a computation. This is more of a kind of a state. How am I doing in relation to the world? This is what feelings are. They're the state that you're in, kind of real-time feedback on state in a language which is not the language of computation. And I think it's the language of mitochondrial fields, which is basically telling you how am I doing in relation to the outside world, in relation to the homeostasis of this bit of the system, and how those bits then the fields link up and strengthen each other and give you a kind of an integrated feedback on right now.
Yeah, I mean, this is, I think, one of the best explanation that I have read for consciousness, in especially thinking from the biochemistry point of view. Um, and so one thing which also came when I was talking to Michael Levin about reductionism in itself, right? Like how deep we can go when it comes to these kind of electrical fields, etc. Because I asked him, like, if we go at the receptor level, we should be able to then get some information out that how these like how probably those single receptors, they are contributing to electrical fields. But then his idea was that maybe after like at a certain point we can't like reduce the system anymore. This is something
I think
emergent.
it's almost the opposite of reduction, which is to say it's an integration. It's I say we have a billion metabolic reactions happening every second. And if you were to shrink yourself down to the size of a molecule, and I finished the book this way, to the size of, let's say, an ATP molecule or something, and then imagine the cell. The other side of the cell would be like the other side of London from where I am at the moment, 20 miles away over there, and there's other molecules like people, if you like, over there. What is going to unite the people over there doing something or other with me over here doing what I'm doing? How do I know now what they are doing over there? Because I do, because the way that a cell works, the metabolism is kind of unified pretty much across the cell. Well, if you're dealing with the Krebs cycle, and the Krebs cycle is involved in pumping, and that produces a charge, and that charge is across the entire membrane, and that's generating a field, and that field is affecting the way that molecules behave, then we have an answer. The answer is easy to say. It's much more difficult to demonstrate, and there's an awful lot to do with the structure of water that we don't understand very well and how the fields might work. But the real key point is that it's not about one pump. It's about the charge on the entire surface of the cell, and how that changes with the input of all the different things that are all directly inputting into it. So it's giving you a kind of an integrated feedback on how all of this metabolic system is doing right now as a single unit, as a single cell with a charge on the membrane, and it's saying I'm all right or I'm not all right. And that seems to me to be the kind of the simplest imaginable unit of what a feeling is. I feel like I'm all right, or I feel like I'm stressed, as a cell. So I don't think that trying to measure it at the level of single pumps, single proteins, is the right way of seeing it. It's really about how do you integrate a whole load of individual entities of molecules, of proteins, of genes, and this entire network that of systems regulation. How does this system know what that system's doing? Well, here's a way of, you can't pump against a high membrane potential. There isn't the energy to do it, which means this system has to close down, and it's more or less keeping everything in the same phase.
Yeah, and about the consciousness part, so how this anesthesia affects membrane of mitochondria or bacteria, for example. Are there any experiments?
Well, there are for simple things like flies, even paramecium and plants, I'm told, can be anesthetized as well. I don't actually know how you can tell if a plant is anesthetized, but um, bacteria, I imagine it's possible to do so as well on exactly the same grounds, but I'm not aware of anybody who's gone out anesthetizing bacteria. Maybe that's what I should be trying to do. Um, and I think it's very, I've noticed that a lot of people get quite angry about some of these ideas, and I think they're misunderstanding what I'm trying to say. And some people are going to be angry anyway because God did it, and any attempt at a materialistic explanation of consciousness is going to offend some people. But other people think that, oh no, I've got a very naive view of what consciousness is. And I'm not trying to explain human consciousness or the loops of awareness of being aware and so on. I mean, I'm not trying to do that. I'm not trying to belittle the complexity of human awareness and consciousness. What I think the question for a biochemist is what is a feeling? How is it that a depolarizing neuron gives rise to a feeling of love or pain or anger? What is a feeling in biochemical terms? So I can see how a computational system, how an AI type intelligence would be capable in principle of surpassing human intelligence. I don't know if it really can or not, but it seems to be, if we're talking about neural circuitry, neural networks as being the basis for computation, and all our decisions are computational decisions about how the world works, then it's very easy to imagine that in principle it would be possible to design a computer system which is capable of equal processing power, and in probably a parallel kind of way. But it would be very difficult for us to know if that system would be conscious, would experience pain or love or melancholy, or the things that actually matter more to us as humans. It would be a terrible thing if our feelings and our emotions and the qualia uh, in our lives were taken away and we became a zombie. That's, a zombie is one of the most painful words that you can hear. Um, but a zombie, you've not taken away anything. They're incapable of the same informational processing, just not the feelings in the same way. So I think there's this. You can understand how a computational system can do an awful lot of information processing, but to try to understand where emotions are coming from, there seem to be two broad ideas. One of them is it's just an emergent property of a sufficiently complex central nervous system. And I think a lot of the AI community would rather see it that way because they have a romantic view that they can construct a machine that will just come alive in all sense of the word and become conscious and suffer pain and existential crisis, you might say. I don't know if we want an AI to have an existential crisis, but And it may be true. I can't say it's wrong, but at no point do I really grasp, so what is this thing called a feeling then? And the other one is more a property of physics, a property of matter, some unknown law of physics, and that would tend to suggest that a stone is capable of some really low-grade form of consciousness, or the sun is in some way. And I just find that unpersuasive as well. It seems to me that it's a property of living things. And again, there will be people who say there's a conscious field across the universe, and I'm just lacking imagination, and maybe they're right. But that's not how I see the world. I see the world in quite simplistic terms, and I tend to think that I think dogs are clearly conscious. I think that elephants are, and octopuses are, and I can imagine that a fly is, and not to anything like the same grade, and not with self-awareness, and not with most of the things that we would think of as human consciousness. But is it possible for a fox to experience pain? Yes, I think it is. I think that's a meaningful concept. Is it possible for a Drosophila to experience pain? Probably no longer in a way that we would understand it, but yes, I'm sure it's possible for a Drosophila to be less happy than another one, to have some unpleasant feeling. What about bacteria then? The further away we get from a complex organism, the less we can relate to it. But is there a difference between bacterium, which is clearly doing well and with a nice buzzing membrane potential, and one which has just been infected by a virus and is deciding to pull the plug on its membrane potential? I could imagine that in terms of fields, in terms of the integration of the cell as a unit, that yes, that would feel like something. I'm feeling well, or I'm feeling ill, in effect. So in terms of this central biochemistry question, and neuron depolarizes and gives rise to a feeling of something, what is a feeling in physical terms? My answer to that would be it's to do with the fields which have been selected over billions and billions of generations, going back to the earliest forms of life, to signify your state in the world in relation to the external world. Am I doing all right, or am I, as a kind of an entity, doing badly? It seems to have selective utility that way. I can imagine natural selection acting on this unit of the cell that is capable of altering its behavior as a result of sensing the environment through a system which is integrating the cell as a unit. I can see all of that in quite materialistic, selective terms. Doesn't mean any of it's right. It's just a way of seeing the question.
Well, I think it's a great way, because if we are saying that there are 4 books written with the title "What is Life," there are 60 books written where the title is, or at least the word consciousness exists in the title. So, and
Probably
still we
hundreds,
don't,
yes.
and still we don't have, I think, a clue to like how to really address the question. So it is like
Yes,
a
I mean,
hard
I think the
problem.
thing is that everybody means something different by the word, and everybody focuses on something different, and it's almost infinitely complex. And it's very easy to focus on your own view, and I am focusing on my own view as a biochemist about what is the simplest possible unit of feeling, and that seems to me to be a coherent story. But there's a phrase I use in the book as well, that there's many a beautiful idea is being killed by an ugly fact, and there are plenty of ugly facts around. And actually, one of the nice things about writing books and being in the lab is that specific idea, you have a general approach. Energy is important, and I'm sure that is broadly true. But beyond that approach, there's lots of little predictions, and those little predictions, you're testing them all the time, and half the time, more than half the time, I'm wrong. It's very humbling. It's very, it's grounding. You constantly finding that what you thought was the right answer here is actually the opposite. It doesn't mean the whole idea falls down, but it means that you've got to keep thinking, you've got to keep rethinking. You're now in this corner over here. Can I get out of the corner, or do I throw in the towel and say the idea was wrong, the hypothesis was wrong? It's great fun being wrong, and it's really constructive because it forces you to keep thinking. And doing experiments and trying to test things means that you're constantly being wrong, you're constantly having to change, you're constantly having to rethink and to try and puzzle out something's not as I thought it was. I'm too simplistic, I think the simple things, it turns out they're wrong. So I try and think, well, is the whole idea wrong, or is there a slightly more complex way of seeing the same thing that explains it? And that's how you make progress. Whereas if all you
I think that's the best explanation of scientific method, that being also aware that you are wrong, but admitting it, that, that you're wrong, and correcting yourself to kind of with the
I think
present
it takes
evidence.
some time to live with
Yeah.
that, and in some way, the more established you are, perhaps it gets easier to say I'm wrong about this and I'm wrong about that. But in another way, maybe not. The bigger your reputation, the harder you fall, you might say. So, I think it's really important for all scientists. I think this is perhaps the single most important thing about the scientific method, insofar as there is one at all. It's that you test things, and when you're wrong, you're the first person to say, I'm wrong. And especially if you were, people get passionate about things and say things they perhaps would regret having said. And then I think you should also be the first person to say, I'm sorry, I got that wrong. It would have been nice if there was more of that during the COVID time. We tended to shout at each other and not apologize when we were wrong. I think it's, science as a method should be objective. We have to have imagination. We have to have creativity. We have to be wrong. We have to test these ideas, and we have to be the first person to say, well, I screwed that one up.
Yeah, exactly. So when we are talking about these ideas, the one thing that I've been thinking of, so in general, I mean, of course, in biology, when we are moving, also in chemistry, a lot of understanding that it comes from physics as well. And our current biology is based on classical mechanics, if we think of. And I mean, of course, now what is going on in physics, especially with the quantum field theory, etc., it's another level.
Absolutely, yes.
But we need to kind of also, especially as biochemists, we need to address that question. I mean, the last decade marks, again, the first book on quantum biology by Jim Al-Khalili, and I forgot the other co-author.
John Joe
The
McFadden.
name exactly.
Yeah.
And so, what are your thoughts on thinking of these kind of electrical fields and quantum biology or quantum field theory? Do you think that we need, like, somehow we are still limited in our systems, and we need to evolve our systems with the updated physics somehow?
I think it's a tricky question. I mean, I am certain that there's a whole language of biology that's linked to electrical fields. I think that's unequivocal from Michael Levin's work, but other people's work as well. And I don't think we really quite have the kit or the vocabulary to understand it yet. But it's also, there's lots of knowledge of birds using electromagnetic fields to navigate, of bacteria do the same kind of thing using magnetosomes. We know about fish and electric currents and electric eels. There's in zoology, there's a lot of knowledge and awareness of the role that electricity plays in biology. And Peter Mitchell and Jennifer Moyle and the mitochondria, we've thought in terms of an electrical potential on a membrane, but not in terms of electrical fields or electromagnetic fields so much. And that was partly because it requires a very specific morphology. If you've got two crystal and they're set at an angle to each other and they've both got oscillating electrical currents going on, then the fields would interfere with each other. So it requires and cancel each other. So it requires quite specific morphological structures for any of this to work. But before you can even begin to look into those things, you have to have an open enough mind to say, well, okay, fields could be important, should be important. But the irony there is, fields go back to Faraday. There is not really about quantum mechanics. There's lots of things which are linked to quantum mechanics in biology. Respiration itself, I talked about electrons hopping down the respiratory chain, like frogs hopping from a lily pad to a lily pad, but actually they're quantum tunneling. And the distances over which the redox centers are separated has clearly been selected. It's rarely more than about 10, 12, 15 angstroms, and that's the distance that allows quantum tunneling of electrons. I think there's whole areas. How many redox? Complex 1 is a strange thing. There's 9 redox centers, iron sulfur clusters in complex 1. Why are there so many? Nobody really knows the answer to that. But if quantum tunneling is real, which it is, and if an electron that sits on this center has a certain probability of being here, and if it's there, a certain probability of being here or there or wherever else, then it's effectively there's a resonance space where that electron could be anywhere on that chain. And that has to affect the likelihood of another. If this electron is here for sure, then putting another electron here is going to be harder than if that electron is smeared in space between all the centers. So you can have a redox tuning of a whole, and this is an area of biology. I'm just saying it may be possible. I don't know if it's real or not. I raise it with people who work on bioenergetics regularly, and we talk about these things, and it's not beyond the bounds of possibility. Is that quantum? It probably is, but it's somewhere between classical and quantum. I'm just reading a really good book at the moment by Philip Ball, called Beyond Weird, I think it's called. And he is talking about the influence of the environment on decoherence. And it seems that these ideas that he's talking about, the book's a few years old now, but it seems that there's recent work. He mentioned it to me, and I don't know exactly what he has in mind, but it's the role of the environment in producing decoherence and effectively the collapse of the wave function. If that's true, the environment is a really difficult thing to get rid of in biology, and that, to my mind, would probably limit what quantum effects are possible or not possible. I don't doubt that they're real and they're there and they're used by biology, but I think it was probably easy to just say, oh, anything's possible, and quantum. It tends to be one of those words that people jump on and then sit smugly on, thinking anything's possible now as a quantum. So it's. I think we need to be rigorous about it, and I think we have a lot to learn about how these systems really work. I think fields are somewhere between quantum and classical, and I think they are real. They don't suffer from decoherence problems, but they have other issues, which is to say they need to reinforce each other to have any chance of signaling over reinforcing each other and signal to, say, the plasma membrane, which is quite a way away if we're thinking about the distance over which electromagnetic fields are operating when we have so few charges involved. So everything I'm saying, I think we don't know the answer to. And I think biology is thrilling from this point of view. It's the intersection of physics with biology, where we just don't know most of the answers at the moment, and we know some stuff, but there's a whole world out there, which is a wonderful world, which we know very little about yet. And that's why I say I think the 21st century is going to be the biology of fields. I think that's the direction of travel.
Definitely. And actually, I've been thinking of, a lot of chaos, randomness, luck, chance, these things that we call environment, that we call, we consider it, but not to that extent, especially in biology. And then again, linked to complexity and all these factors, we need to, I think, also address these kind of issues, right? Like how these chance events occur and like what is their contribution in terms of building up these systems, etc. And I was thinking that maybe it's limited somewhere in the chemistry, but I don't see any also like something interesting coming out of those explanations. So the only explanation that I could find is probably at the physics level, which can really change our understanding of biology. But I don't know, again, as you said, that these are all speculations.
Yes, but I don't like the word speculation very much. It's true, but people use the word speculation to dismiss it as hand waving and basically lacking in any kind of scientific substance. And I would say that a hypothesis is a speculation. A hypothesis is a flight of fancy into the unknown. But what makes it science is that you name the steps and you say, if this is so, then this should be so, and I can test it like that. And then it becomes science. And this is something that also applies very strongly to the origin of life. You need to name the steps and you need to test them. And where we have a long continuum from very simple prebiotic chemistry all the way through to complex molecular machines and so on, it really matters what's the order in which you get there from here. what comes first, genes or metabolism and so on. So specify, what do you think happens, and then how does it get to the next step, and what's the next step, and how do we test those steps, and how do we put all this together? um, very different predictions come out of it. You could say the whole lot is speculation, and in some sense it is, but it's also a hypothesis. I think we're a long, long way from creating life in the lab, personally, in terms of life that anybody who uh, just had a kind of an unbiased view of what a living system actually is would recognize as life. It may be that someone can say, "Oh, I've created life in the lab," and what they actually mean is they've got something which is capable of making a rudimentary copy of itself and growing a bit. And most people say, "Well, it's not alive, is it?" So to come up with something resembling a cell, I think we're a long, long way from doing that, and I don't think there's really any danger of it happening in my lifetime. But to understand how life could have emerged, to piece together a coherent story that intellectually goes from one step to the next step to the next step, all the way through to cells with molecular machines and genes, and to have tested virtually all of those steps and demonstrated that under some set of conditions it really does work, which then turns it from just speculation into, let's say, a hypothesis that's got some meat on the bone, that might be true because you've demonstrated that the individual steps are possible. Doesn't mean that they can all join up together and do what you said they could do, but it just means that intellectually I can begin to believe that this is not just a piece of make-believe. This is not a figment of my imagination, but the individual steps are possibly real. And I think that's kind of the best we can do, in a way. Maybe people might find that disappointing, but I find it quite thrilling, because what it means is that I think we can understand intellectually, with enough substance, to say that everything I'm calling on here is possible and is real and is not a flight of fancy. How a non-living, sterile, inorganic planet can give rise to the living planet that we see around us. It's a wonderful challenge to try to explain, to try to understand. I think the best we can do is to have some kind of sketchy understanding of how it came about that makes coherent intellectual sense, which has been tested and demonstrated that is not incoherent.
So I had another conversation. This was with Eddie Pross again on
Okay.
Origin of Life, and when I asked him, okay, what are we trying to do here when we are trying to address Origin of Life question, and his idea or his explanation was that we are just looking for chemical laws, chemical and physical laws, which can give us an explanation, or which will tell us simply that okay, if we follow these laws we can create kind of this synthetic life in the lab. Again, it's abstract, but
Yes, I mean, that's not a million miles away from what I just said.
yes,
And yes, in principle, if you follow them, you could do that. But in practice, um, the kind of what you can do in a lab, you can test one reaction and you can test it over a short period of time. You can't leave it for extended periods of time, and you may get the conditions a little bit wrong, or the ions. We're always simplifying a complex system. And you think about the kind of hydrothermal vents that I'm really talking about, and they're on a large scale. They can be 60 meters tall. They're networks of interconnected micropores with fluids continually moving through them, with complex chemistry, all kinds of mineralogy that's in there. All kinds of complex chemistry in the oceans, all kinds of ions that are in there. And 4 billion years ago, before the crust had differentiated from the mantle, probably the whole seafloor would have been doing this kind of chemistry. So we have a planet-wide laboratory where the chemistry that's happening is broadly similar but differing in every place in many chemical details over as many million years as you want it to happen. I would rather think in terms of nanoseconds and things, or seconds, rather than millions of years, because it's chemistry and it has to happen fast in my book. But the idea that we can spend a few years in a lab messing around with these things and come up with something like that, it's ridiculous. But can we demonstrate in principle that this kind of reaction that we think needs to happen here really can happen here? well, then it begins to give some substance to the belief that this kind of chemistry is possible. And then you think about, well, a global scale of a laboratory doing this kind of chemistry, and you can at least imagine that some chemistry that I struggle to make happen in the lab a little bit can be happening on industrial scale here,
Yeah, I mean,
planetary scale.
yeah, according to Lee Cronin, he would say that we want to literally convert sand into something living, which
Yes, but I mean, Lee Cronin is fantastic. He's got amazing ideas. Um, I don't know if any of them are going to work, and I don't know. And he would probably say the same thing. He wants them to work, but does he, in his heart of hearts, really expect them to work? But even if they do, um, would most people recognize them as living systems? I think perhaps part of his point is they wouldn't and they shouldn't, because there's a different kind of living system that's not based on carbon or not based on life as we know it. It's using the same principles rather than the same chemistry. But I think a lot of people would struggle to recognize it as living, as life. He can come out and say, "I've created life in the lab," and he'll show some system. People saying, "Oh, really?"
yeah,
If he does it, then he can explain to us. But I think he started out as an inorganic chemist, trying to make life purely from inorganic molecules. I think
yeah, using
that he's
molybdenum or
added
something.
carbon into his systems more recently, and that to
Yeah.
me suggests that the purely inorganic system doesn't work quite as well as he might have hoped.
Okay. Yeah. Then maybe I should have
I
one
mean, maybe
conversation with.
yes. I mean, I've actually never met Lee Cronin.
Oh,
I don't know how
interesting.
not. I mean, we've exchanged emails, and we were both on a radio program together at one time. But I admire his work,
Yeah.
and actually we have some convergence. But we're starting in very different places. I'm basically starting with carbon-based life as my guide to the principles of life, and he's starting with the principles of life in a very abstract sense, compartmentalization, selection, the kind of things that
Yeah.
biologists would talk about, but then
Yeah.
saying, how far can I do this with a completely alien, not carbon-based life system, which is capable of being driven by the same processes and giving rise to something which is a living system, but just not as we know it. It's an admirable thing to do. And I think he's moving in some strange way. We're probably moving closer rather than further away from each other, because if he's beginning to introduce carbon into his system, I think perhaps he should introduce charge into his system. I don't know if he is doing
Well,
fields.
He and Sarah Walker, they've been developing this theory, assembly theory, which
Yeah,
they call
yeah.
it, and this is what exactly they are trying to explain, that how these molecules, they can
And
evolve.
that's
Yeah.
I mean, I think that's a perfectly reasonable thing. There
Yeah.
are a lot of steps in
Yeah.
any biochemical pathway. Um, and but there's some degree of ambiguity about it, because what they're effectively saying is, if you've got an index of more than, I don't know what their actual numbers are, but effectively, if you've got to have more than 8 separate things
Steps.
going on
Yeah.
here, then it would be a living system that gave rise to it. And if we can detect something that requires at least 8 different steps to make it, then we can be fairly sure it was made by life and not made by a non-living system, which is only capable of doing, let's say, 3 or 4 steps. That's probably a dreadful um, oversimplification of what they're doing, and I would broadly agree with that. And what's very neat about it is it doesn't specify that it has to be carbon-based. It means that you can keep an open mind about what other forms of life might be out there, which is better than saying, "Oh, I'm looking for a chiral carbon molecule," or some people say, "Let's find ATP," which is madness. Why would you find ATP? Um, the problem that I have with it is from our own work. If you have a non-living system which doesn't have any genes in it, let's say, I've got membrane heredity, I've got CO2 fixation taking place across a membrane, and I've got a spontaneous proto-metabolism taking place in the absence of genes or enzymes. The kind of the core biochemical pathways of cells are spontaneously happening there. And I can imagine that I might get trace amounts of nucleotides in this system. And already that is a big ask. And there's people in the lab who are working on trying to make nucleotides and have had some partial success. But you can imagine that it might be possible to make a bit, but we need a lot. If you want to start polymerizing to make DNA or RNA, we don't want trace amounts. We want bucket loads of these things, which means these systems have to get better at making them. Otherwise, the whole hypothesis is wrong, which is perfectly likely. But if it's not wrong, then the requirement is that this simple system has to be capable of driving its own biochemistry through positive feedback loops and autocatalysis and perfectly reasonable processes to get better at making complex molecules. So to make purine nucleotides like ATP, for example, we're talking about 12 dedicated steps from starting with phosphoribosyl pyrophosphate, for example, as the starting molecule, which we're already dealing with another 6 or 7 steps to make that from CO2 and hydrogen, more than 6 or 7 steps. So we're up to 20 steps here. And this is a system which is, what I'm describing to you is effectively a non-living system capable of a rudimentary non-genetic form of heredity, which has bootstrapped itself up from a simpler system through positive feedbacks. Now, all this may be complete make-believe on my own part, but what it's effectively saying is you can get to some quite high levels of complexity that we would immediately associate with life through a non-living system that's bootstrapping itself up to the level of a living system. So there would be ambiguity in my book there.
Yeah, and I think that does make sense, and we'll see that how far we can go with it. And I think more of your work will also highlight that how much we can understand with the just starting with hydrogen and carbon dioxide, and how far we can go with the replicating or producing the components of the life forms that we have now. So are there any possibilities to reduce this system? So I mean, of course, as you said, that there are 6-7 steps in the Krebs cycle, and if we can kind of reduce that to 2-3 steps instead of
Well,
something
we
like
don't
that.
need a whole Krebs cycle at the beginning. What we really need it to do is to make the carbon skeletons that are used for the rest of biosynthesis. And most of the amino acids that are the most ancient amino acids are 2, 3, 4, at the most 5 carbon atoms long. And they're all derived from carboxylic acids. So Krebs cycle intermediates, either directly or indirectly. But so what we need the Krebs cycle to do before it becomes a cycle is a kind of a linear pathway of effectively adding CO2 onto carboxylic acids. That's what it's doing. And what that, if you've got a carboxylic acid, so I don't want to get too technical about this, but you've basically got an end group which is effectively CO2. It's a carbon atom with 2 oxygens attached to it and a negative charge on it. And we need to add another CO2 onto this. So we take out one of the oxygens and put another CO2 on there. And now we have a double bonded oxygen on what's called the alpha carbon. And that is quite reactive and will tend to react with nitrogen. And then you have an alpha amino acid, which is exactly the kinds of amino acids that we have in life. So building the right amino acids, we need the carboxylic acids, and we need to extend them by adding CO2 onto the end of them. And this way, starting with these simplest building blocks, we get biochemistry as we know it, including alpha amino acids rather than any other kind of amino acid, which in principle you could have. And so we don't need a complete cycle. We just need to be extending up to C4, 4 carbon units, 5 carbon units at the most in there. And very often, most of life is not running a complete Krebs cycle. Most of the time, it's a partial cycle, or it's 2 halves, or it's just a bit here and out there. And this idea of we always have a Krebs cycle, which does a complete spin of a cycle at the heart of all of life, is a kind of Platonic ideal, which is just not true for most things most of the time. But we love cycles and circles, and it's we've deluded ourselves by taking it too literally.
Circle of life, as we call it. So
Yes, we love the circle of life, but this is more of a mad roundabout at the heart of things, and the traffic can go both ways through it.
So in the book, you mentioned that the next book would be how to get information from metabolic systems. Is it right, or you're just thinking about it?
Well, I'm not going to write anything else for 5 years. It's exhausting writing a book. It really takes a lot out of you. It's wonderful fun, but the next book will probably be on the origin of life. And the second reason I want to wait for 5 years is that I honestly think we'll have made a lot of progress over that 5 years. I think that I don't imagine that most of the world will agree with my viewpoint on it, but I would like to think that if the work that we're doing at the moment keeps on going in the direction it is, that in 5 years' time I will have some kind of broad picture of here are the steps that need to happen to go from a non-living planet to genes and molecular machines. Here are the bits that are still missing, but these are the bits that we think it can work this way, and effectively join them up. I've written several books where there's a chapter or even 2 chapters on the origin of life, but I've never written a whole book on it. And it's only it's not the only thing I'm working on, but it's the thing that I think is too exciting and too close to not do the whole job. What I've written about before is always focused on one aspect of it, and I've never addressed information in a serious way. So the next one, yes, will be the origin of life right through into information, and how molecular machines are coded by information and how they start.
That's just fascinating. And then after another 5 years, it'll be a book on consciousness, I
Who
hope.
knows? I don't.
Yeah,
I think even looking 5 years ahead is probably quite a long way. Maybe I have
yeah.
a heart attack tomorrow, so I don't want to get ahead of myself.
No, I mean, now you can, with exercise and stuff, now you
Yeah,
can at least
I shall go
maintain
for a
some.
run right after this. I've
Exactly.
not managed to go at all this week, so I really need to get some exercise.
Exactly. So it's really exciting to know your work, and I think we'll be waiting for more of the stuff coming out of your group, from your ideas. Uh, so
Well, increasingly, I have to say that the people doing PhDs and postdoc work with, I'm not going to name them all here now, but they're having more and more ideas themselves, and I'm feeling more like a passenger sometimes certainly than I used to. I'm used to having all the ideas and saying how about we test this, and I'm finding increasingly that people are coming to say, well, how about this as an idea? And I think, wow, that's a really good idea. It's great fun,
yeah,
um, being surrounded by young, imaginative, scientific minds of people who are thinking about the most exciting questions that we can have as a human being, as a scientist. And it's a wonderful thing to be able to um, have a lab that for now, for the next 2 or 3 years, and after that, who knows what happens because funding runs out and you're always scrabbling around. But for now, it's an environment where people are free to think and experiment and pursue their dreams. And I think in some strange way that maybe of all the things that I do, this is the most liberating of all of them, is that you give people the space to find themselves.
I think sapiens have been creative for most of their times. All we require good mentors. I think that we just need some of the
Yes.
like people who can guide them well, and
And I
I
don't
think
think there's anything more noble in science than being a good mentor, than
yeah,
helping people to find
exactly.
themselves and develop themselves. I think if I aspire to anything, that's
Yeah.
probably the thing I aspire to most. And you're always very aware of your own failings in where you could have helped someone better or more, or I don't know. But at least if you try and think that way, you try and get it better next time. I like to think that anyway.
Yeah, and I think this is already a profound message, not only for scientists and academicians, but also like for the society, for people who that just guide people in better way, and it's
Yes,
just
I mean,
amazing fun.
I think I'm far from alone in being very troubled by the deep splits in society these days. And there's a deep distrust of science and scientists and rationality even, um, and a strong feeling from a lot of people that scientists are a kind of priesthood who preach at people and tell them how to behave and what to do and don't like it. I think it's a deep human reaction to being told what to do by someone who seems to be holier than thou, um, you know, sees themselves as superior in some way. And the whole setup is toxic, actually. And it's not what science is really like. Science is very creative. And there is really not much of a hierarchy, that ideas come from anywhere, and it's not nearly as representative as it ought to be, but it's more representative than it used to be. And again, I think one of the things that I really want to try and do is open up science to everybody. Um, and this idea that you've got to do well in exams, even that is a questionable thing. I think, yes, there are some people who are very good at doing exams, and they may also be a very good scientist. But there are also people who are terrible at exams who may be a brilliant scientist, because they think differently. They think their own way. They cannot respond to being told, you go away and learn that and then write an exam and get everything memorized and back on the page in 40 minutes flat, otherwise you fail. So we drive people out of science that way, people who have often creative imaginations, who see the world in a different way. And the more that we embrace people who have their own way of seeing things and help them to find the best form of themselves, and embrace a much wider cross section of society as can be a wonderful scientist, can make a real contribution. And it seems like a world that should be real and yet is so far away. And I don't know how we get there from here, but I think we have to try.
Yeah, we do have to, and especially with all the misinformation, etc. Somehow we need to balance out. We need to balance out the opinion of experts, but also somehow that we don't go too strong on people uh, giving our opinion and understanding.
Yeah, it's a tough one,
Yeah,
because
it
experts
is.
are wrong about a lot,
Exactly.
but they also know a lot about a lot.
Yeah.
So it's a kind of a difficult balance, and maybe I was talking about the importance of being wrong on a daily basis in the lab. It prevents you from becoming too full of yourself as an expert. I know what's going to happen. Well, no, I know every day that I get stuff wrong. But I also spend my whole life thinking about these things and wondering about it and worrying about it and trying to work out where I went wrong and how I get better at it. And there's uh, you shouldn't believe what I say, but neither should you dismiss it completely, because it's not something that I just thought of 2 minutes ago and kind of shouted from the rooftops. It's stuff that I spend my life thinking about and worrying about, and it doesn't mean I'm right, but it probably means that you shouldn't dismiss it completely immediately either. Um, Less quickly than you might dismiss someone who didn't even know anything at all about it and then has a very strong opinion. So we need to find a balance that recognizes that it's not so much, it's more a case of how long have you spent living with these ideas, how much do you wrestle with them, how much do you care about it, and where are your biases, and where are you just wrong, or where do you not see it from someone else's point of view, where they're, I was talking about outsiders coming in often have the best ideas. The longer you're in a field, the more stale you become, and the outsiders who have been there, all of these things contribute, and it means it's difficult. And expertise shouldn't be dismissed, but neither should it be seen as the answer to everything.
Yeah, also I think it depends on what kind of this pre-biases that we have, or like in Bayesian kind of understanding. Um, so if we have those kind of factors, or that kind of thinking already, it depends. So sometimes we align with people so easily, but then sometimes it's so contrasting the difference. So
Yeah, yeah,
and that's difficult to cross that barrier until
yeah,
unless you literally try to educate yourself and try to understand the perspective. And as you mentioned in the book, be a molecule in that cell, just like, if you want to understand something, like, so yeah,
yeah,
that's the
yeah.
interesting part. So with that, I think this was a great conversation. We discussed a lot
I've really enjoyed
about
it.
origin of life, bioenergetics, your work, which can actually transform health in this way. This was also another important topic that we didn't cover to that extent, but a lot of people are already talking about it. So that was also kind of refreshing for me to read, and I was really inspired by it, thinking
Thank you.
about biochemistry and biochemists talking about all the things, and doing
I have to
this
remind
sort of
you, you're
great
doing a
job.
PhD in biochemistry, so if you didn't enjoy it, then who the hell would?
Exactly. I did. I thoroughly
I'm
enjoyed
glad
it,
you
and
did.
I told you that I enjoyed the book. I enjoyed your work, your talks. I've been listening to you on other platforms. It's been amazing, and yeah, this thinking is, I think, kind of transforming, or it will transform. I hope that we'll go a little bit beyond this information based
I hope so.
research. I hope. Yeah.
I hope so too.
Yeah.
Great.
So thank you so
Thank
much
you.
for
Thank you.
accepting the
Been a pleasure.
invitation. Thank you.
Bye.
Bye.